Hello, my name is Vanessa Orduno and my partner is Sebastian Pennant. So our project is based on the reconstruction of the Spanish flu. In 1918, the influenza pandemic was the most severe in history. It was caused by an H1N1 virus with genes of avian origin. It spread worldwide during 1918 to 1919. It's estimated that about 500 million people became infected and about 50 million died. So signs and symptoms of the Spanish flu. So what was different about the Spanish flu from other influenza pandemic? Well, it was the mortality rate of young and healthy individuals. The flu was most deadly to 20 and 40-year-olds, having a mortality rate of 2.5%, whereas influenza usually has a 0.1% rate. Many people developed symptoms and died within a couple of days. The average infected patient would develop normal flu symptoms such as fever with nausea, diarrhea, sore throat, and chills. The patient's cough would drastically become worse, and they would develop dark spots on their cheek that caused them to suffocate as soon as a couple of hours. After the pandemic in 1919, it stopped. Most who got infected died, and those who survived developed immunity. Throughout the years, there have been other types of influenza pandemic, but none as deadly as the Spanish flu. Today, scientists have been on the hunt to find out what made the disease so deadly with newer technology and what they can do to combat the next pandemic.
Many attempts to find a live strain of the virus have been unsuccessful due to the fragile genetic material of influenza. However, in 1990, Johann Holton, a scientist at the NIAID, discovered a live strain of influenza. It was found in a mass grave in a remote village near the town of Brevig, Mission, in Alaska, under the permafrost of an Inuit woman buried more than 6 feet under ice for over 75 years. The cold conditions and woman's ample fat storage kept the virus alive in her lungs long enough for scientists to recover viral RNA and attempt to sequence it. Scientists were able to use the viral RNA fragments and sequence the viral genes using reverse transcriptase. They found 8 genes that made up the main components of influenza. It took scientists 9 years to complete the coding sequence of the virus. Once the full sequence was in hand, other researchers set about reconstructing the 1918 virus. They stitched the 8 genes into a regular flu virus genome and shipped the DNA to the CDC. There, virologists inserted the DNA into cells to make a live virus. They infected live mice with the 1918 reconstructed virus. 3 out of 5 mice died during the experiment, but they found a couple of interesting things associated with the mouse's lungs. A group of 3 genes enabled the virus to weaken a victim's bronchial tubes and lungs and clear the way for bacterial pneumonia. Other than that, severe tissue injury, oxidative damage, and an increase in murin genes involved in apoptosis were all found in the lungs. This told scientists that most people died of pneumonia and respiratory failure caused by Streptococcus pneumoniae, S. aureus, and S. pyogenes, not necessarily influenza. They found that the 1918 influenza virus had the ability to rapidly cause extensive damage to the respiratory epithelium and spread at an incredible rate. On closer examination, they found that influenza can infect many kinds of cells, besides lung cells, because it doesn't rely on an enzyme that is only present in specific cell types. Finally, scientists found that genes found in a bird flu were similar to the 1918 flu. The purpose of reconstructing the Spanish flu was to get a better understanding of the deadliest pandemic in history. The biological properties and virulence of the pandemic influenza virus were poorly understood. Reconstructing and experimenting on the virus is vital to understanding the disease process and can provide important insight into the basis of virulence. This kind of information will enable us to devise appropriate strategies for early diagnosis, treatment, and prevention should a similar pandemic virus emerge. With how global our world is, it is impossible to isolate a region. Such research will provide us with general principles with which we can better design antiviral drugs and other interventions against all influenza viruses with enhanced virulence and maybe have the ability to produce vaccines capable of handling the mutations of the next pandemic influenza virus. Thank you, and I hope you enjoyed.